All right. So we'll continue from where we left off. So I just want to talk about how exactly is this peptide bond formed. So it is very important for you to get this right. So start by drawing the amino acid itself. Okay. So this is the chemical structure. So we have H there and the amino group. So what we meant by saying the carboxylic group of one amino acid is going to react with the amino group on the other amino acid. So draw another amino acid right here. Okay. Okay. So what happens is that the H molecule, please take note, the H or the hydrogen molecule on one amino acid reacts with the hydroxide group on a different amino acid, on the carboxylic group. So when these two mix, water is going to be released in the process. Okay, so this is called dehydration synthesis. Okay, dehydration synthesis. Okay, because what we're going to form at the end of the day is a peptide bond. So this water is going to be lost in the process. Okay, so let me repeat this part. So this is to tell you that the hydroxide group from the carboxylic group of one amino acid reacts with the hydrogen group on a different amino acid, and hence water is released in the process. Okay, and it's called dehydration, or rather condensation, where water is now being released. Alright, so let me now show the actual peptide bond that is going to be formed. Okay, so this is the bond that is going to be formed as a result of the condensation reaction. Okay, so remember that we have two amino acids here that are involved in bond forming. So this bond that is right here, oh sorry, there's another group here. Okay, so this is the R group, this is the H. Alright, so what I want you to see is that this bond, let me just use a different marker. So this bond that you're able to see right here is what we are calling a peptide bond. Okay, this is a peptide bond that has been formed. Alright, and what has been released. So when we have peptide bonds that are between two amino acids, okay, we have a peptide bond between these two amino acids. We also have what we call dipeptide bonds when we have two amino acids. Actually, two amino acids is called a dipeptide bond, right? Alright, so let me just highlight one more thing. So when we have so many amino acids that are being formed through these peptide bonds, we call them a polypeptide bond. Okay, that's what we call it. So the next thing I want us to learn is how these amino acids actually form these polypeptides. Okay, so we'll look at the levels of protein structure. So levels of protein structure. Okay, so when we look at the levels of protein structure, we're just talking about proteins can be found in different types of shapes, and when they're found in a certain shape, there's a way in which they're able to react, okay, and become more functional. So the first shape that we're going to talk about is called the primary structure. Okay, so when you talk about this primary structure, it is simply the number. So the primary structure includes the number and sequence of amino acids. Okay, so these amino acids, remember, they are held together by peptide bonds. So the primary structure will simply tell you the basic structure before these amino acids even begin to fold themselves into different shapes. So if we have this amino acid here, an amino acid there, and another one, and these are linked by a peptide bond, this structure is what we call a primary structure. Alright, it is simply the number and the sequence of these amino acid. So you need to note that each protein has a unique sequence of amino acid. So what happens is that, for instance, if here you have valine, maybe here isoleucine, you have threonine or tyrosine somewhere here, the way these amino acids are arranged would determine the shape and even the function of the protein that we're talking about. So each protein has a unique sequence of amino acid. So the first protein to have its amino acid sequence actually elucidated, or rather determined, was the insulin. Okay, where it had two peptide chains, in which the chain A had 20 amino acids and chain B had 31 amino acids. So this insulin was the first amino acid, was the first protein, sorry, that was determined. Okay, and remember, I was saying in the primary structure it is basically the number and the sequence of amino acids. Okay, so there isn't so much to talk about here, because it's just like the basic level of a protein. Alright, next thing we'll talk about the secondary structure. So we have what we call secondary structure. Alright, in the secondary structure, a secondary structure of a protein is actually the arrangement. So please take note of this. So it is the arrangement of the amino acids. Okay, the amino acids within the chain. Okay, so within the chain. So what I'm trying to talk about is that when you look at primary, we just have a long chain of amino acid. Okay, they are bonded together. It's a long chain. But when you look at the secondary, it is the way these amino acids are actually connected to each other. Okay, and the way in which they're connected is what will give them their shape. So for instance, we have two types of secondary structures. Alright, just a minute. Alright, so these two types of secondary structures that we have what we call the alpha helix. So we have the alpha helix and the beta helix molecule. So these are the ones, or beta pleated sheet. Okay, or a beta pleated sheet. Alright, these are the types of secondary structures that we have. So note that in the alpha helix, it is a right-handed coiled or a spiral structure. So it looks something like this. Okay, so right here we have amino acids that are actually interacting with each other through hydrogen bonds. So this is to tell you that if there is an N here and a hydrogen, these two are going to bond through a hydrogen bond, and another one right here. So it is because of this bonding that causes a secondary structure. Okay, and to be specific, we're talking about the alpha helix. So the NH group of one amino acid is hydrogen bonded to the carboxylic group, because the carboxyl group is the C double bond O, and because of this there will be hydrogen bond, which will cause the amino acid to actually exist in a coil form. So a perfect example I can give you is keratin. Okay, so keratin is a protein that is found in your hair. So if you notice that your hair is usually curly until blown, of course. So what happens is that this coiling comes about due to hydrogen bonding. Okay, so it is found in hair. These types of alpha helix are found in hair, in wool, in claws. I hope you know what claws are exactly. So a claw is just like the nail of an eagle or a chicken or birds in short. Okay, so birds have claws. So they are made up of these types of structures that we're calling the secondary structure. Then we're going to talk about the beta pleated sheet. Okay, so remember that in this secondary level, I'm talking about the hydrogen bonds that exist between the NH group of one amino acid and the carboxyl group of a different amino acid. That interaction is what will cause the the polypeptide chain to actually coil around itself. Okay, the next one that we're going to talk about is the beta pleated sheet. So we have beta pleated sheet. So what we're going to learn in this is that beta pleated sheets are extended forms of polypeptide chains. Okay, they are formed in certain proteins due to the interference of the hydrogen bondings of the certain R groups. So we know that we have an R group in an amino acid, right? Okay, so this is the R group. So there's a way in which these R groups interact with each other, okay, depending on their nature. So when these R groups interact, they form something like, it looks like a zigzag. Okay, these are what we call the beta pleated sheet. So you find that the R group of this amino acid is interacting with the R group on the other amino acid parallel. So these are called beta pleated sheets, and they're actually more rigid than the alpha beta pleated sheets. So the inability of the amino acid proline to make different interchain hydrogen bonds. Sorry, let me just go over that. So the beta pleated sheet is best described in silk fibrine. So we can find it in silk. Okay, so you notice that silk is not so easy to break because of its nature. All right, so this protein contains a number of adjacent polypeptide chains. So these are they are adjacent because they're close to each other. Okay, these are the polypeptide chains, and they are more extended than the alpha helices. All right, as you can see, they're usually parallel from each other. Okay, they're parallel, meaning they're going almost in one direction, but they will never meet. Okay, so this is what we are able to observe. So these parallel polypeptide chains are actually brought together due to the interaction of the R groups. Okay, this is what causes the beta pleated sheets. So they are brought together by interchain hydrogen bonds between the CO. So CO is the carbonyl group. The NH is coming from the amino group. So they're saying that this interaction is maintained because of these two molecules. All right. So the next type of level of protein structure that we talk about is actually the tertiary type of structure. All right, so we have what we call the tertiary. So we have the tertiary structure. All right, so what exactly occurs in this tertiary structure? Okay, so in this tertiary structure we have a number of bonds that participate. We have what we call hydrophobic interactions. We have van der Waals. We have ionic bonds. We have hydrogen bonds. Okay, so these are the bonds that are actually involved in the, oh, we also have sulfide bonds, not forgetting one of the strongest bonds that exist in the tertiary structure is the sulfide bond. Okay, so what we see here is that the tertiary structure of a protein is actually the bending and folding of the protein. So remember that at first we just have a sequence of amino acids in the primary, but now that the protein is going to bend around itself and configure, this is what we're calling the tertiary structure. Okay, because the tertiary structure is that the protein is able to bend and fold itself in such a way that it forms a globular shape. So this is point one, very important. One, it is globular. It has a globular shape, so it means it looks like a globe. So this is very important. If you want to differentiate this, this is a very important point. The next thing is that it only has one subunit. Okay, so it only has one subunit, and the most common example is myoglobin. Okay, so myoglobin is actually a protein that transports oxygen, and it is usually found in muscles. Okay, now myoglobin is different from hemoglobin because myoglobin is able to store the oxygen, all right, whilst hemoglobin is used to transport that oxygen. All right, so the best example of these are myoglobin. You need to note that. So when these proteins that are found in the quaternary structure, immediately they lose their shape. Even the function becomes distorted. For instance, we have people that are actually sickle celled. They have sickle cell red blood cells. So these are red blood cells that have an incomplete S shape. So if this is a sickle cell, it will look something like this. So because of this shape, you find that it is unable to carry enough oxygen atoms. But unlike this one, which is biconcave, this one can carry excess oxygen, right? Unlike the ones that are distorted. All right. So this is to tell you that when the protein gets denatured, even the function will be disturbed. All right. So the next thing we'll talk about is we were still talking about the tertiary structure, and we note that it has sulfur bonds. This is also very important, and these are relatively strong. These are strong bonds. They're not easily broken. Okay, so in the case of insulin has a three-dimensional structure. It is further stabilized by these sulfide bridges on the cysteine residues. Okay, so just trying to explain about these alpha helix that we're able to see through this bond. So what happens is that we have something like this, right? So what happens is that when these begin to interact, they form what we call subunits. Now, in this tertiary structure, these proteins are globular in shape, so they appear to be a globe. All right. Okay, so the three-dimensional structure is further stabilized by the disulfide bonds in the cysteine. Remember, we said we only have two amino acids that are sulfur containing. That's methionine and cysteine. Okay, those are the amino acids that contain sulfur. The next thing we'll talk about is the quaternary structure. All right, we'll talk about the quaternary structure. Okay, so we have quaternary. Okay, so we have what we call the quaternary structure. So in this structure, it is actually the aggregation. So it is the aggregation. So when something aggregates, it means they come together. So aggregation means coming together. So when you find out that these are the chains that are held by hydrophobic interactions, hydrogen bonds, ionic bonds, they have proteins that are in this structure there, quaternary. So this is to tell that these polypeptide chains, for instance, if this is a polypeptide chain, there's another one here and another one. When these are bonded by peptide chains, this is what we call the quaternary structure. There is more than one subunit. Okay, so more than one subunit. Okay, these are what we call the quaternary structure. The best example we can give is hemoglobin. Okay, now I want to clarify something about hemoglobin. Hemoglobin can also be classified as tertiary structure because of its shape, since it's globular. Okay, but in terms of subunits, hemoglobin is quaternary because it has 4 subunits. Okay, while myoglobin, we know that it only has 1 subunit. So this hemoglobin is able to transport oxygen to all parts of the body. Okay, so hemoglobin has beta chains and it also has alpha chains, so it's able to transport oxygen to all parts of the body. Then another thing, we're actually wrapping up this topic, so the next segment is where you actually ask questions where you're not actually clear. Alright, so this is the quaternary structure. It has about 4 subunits. Okay, and this is where the polypeptide, or rather these amino acids or amino acid sequence, are able to aggregate, okay, and actually form a 3D structure. Alright, so I'll talk about 2 types of proteins. We have one, insulin, and collagen. So I'll just briefly talk about that. Okay, so we have what we call insulin. So insulin, basically, it is a peptide hormone, okay, whose major role is actually to regulate amount of glucose in the blood. So when there is so much glucose in the blood, the insulin is able to facilitate the activation of glucose into glycogen, where it is stored up in the liver and in the muscles. Whereas when the level of the glucose becomes very low, it promotes the breakdown of this glycogen. So when the glucose is high, we have glycogen formed. When the glucose is low, the insulin, there's a hormone that we call glucagon that is able to convert this glycogen back to glucose. Okay, so it is like a chain reaction. They are interconnected. Alright, so insulin promotes the synthesis of glycogen, which is stored in the liver and the muscles. So when the level of glucose becomes very low, insulin promotes the breakdown of glycogen to release glucose. So insulin is active when it is in the chain form. So this is to tell you that when insulin is in this chain form, okay, this is just an illustration. So when insulin is in this chain form, it's able to function well. Alright, unlike if it was in a different shell. And then we only have this insulin that is being used for this conversion. So it can be converted into dimers, or rather 2 polypeptides, Then we have 6 polypeptide granules, depending on the type of insulin that you have. So this is to tell you that we have so here they're just trying to tell us what type of insulin that we have, or what exactly constitutes this insulin. Okay, so we have the hexamer, which is in form of the insulin. So we have something like this, okay, bonded through hydrogen bonds, of course. Okay, so this insulin, as we said, it is a protein hormone which when secreted controls the amount of glucose in the body or in the blood circulation. So when you look at insulin, it has a quaternary structure. So you need to note that the next protein that we're going to talk about is actually collagen. Okay, so we have collagen fibers. Okay, so collagen is a fibrous protein and it has 3 polypeptide chains. So one, we find that it is fibrous. It has about 3 polypeptide chains, okay, that are wound around each other and they form a triple helix. So they form something like this, so that the 3 of them, okay, so these are the 3 polypeptide chains that are fibrous in nature and are the ones that form the collagen. So the secondary structure is the most important. This is to tell you that the secondary structure of it being coiled like this is one of the most important key factors or key elements of these collagen fibers. So you notice that one subunit is made out of a secondary structure, so is the other one and the others that are there. Okay, so it is in the form of these elongated fibrils that I've actually highlighted there, and these they form the fibrous tissues. So collagen can actually be stretched. Okay, it has stretching abilities, it is almost elastic, and it is important for the connective tissue function of the tendons. Okay, we need what we call the collagen even in the cartilage, the bone, the skin, and even the bone marrow. We have this cartilage that is here, as well as the digestive tract. Okay, so this is about the collagen. All right, so this is actually the end of proteins. I'll just stop recording and